rock-based project, which is looking at mineral crust formation and microbes living in those rocks. So we probably pick some of those up on this dive, or this watch. Yeah.
Would anything in this frame of view here qualify for those altered rocks?
Well, yeah, it definitely could. So I was assuming we would kind of move along a little bit and see if we see any life before deciding if we just maybe spend a little time sampling for rocks. So we've gone down maybe about 30 or so meters, I think, maybe a little more, as we've been moving in this northeastern trajectory. Doesn't seem to be a sweet spot depth for much big charismatic fauna. Okay, front row.
Yep.
Now that we've gone down about 2,400 and we're not seeing much down this way.
Yeah.
Maybe if we could start ascending again while we're continuing on this northward trajectory.
Sure. We'll let this move finish out.
Yep.
And something like that on HiPAC if you have it up.
That's a good enough guess, I guess.
Sure.
It's kind of hard to know without really knowing what the slope is going to be like ahead of us.
A lot of these, as you know, but maybe the audience doesn't, a lot of these features, when we do find a wall or something like that, it's not necessarily continuous and we can just follow a strike, and you know that it often shifts and or stops existing altogether. So it's kind of hard to find. And then that's presuming that those are the features that the coral get all of their current and nutrient needs from. So
yeah,
it's kind of a big old guessing game we're playing here.
Yep.
But
I think if we kind of pick that trajectory and kind of head towards what may be that second northern lobe,
sure.
We'll see what we get.
Is there potentially any kind of life that we, is just either burrowed in the sediment or so small that we can't see it?
Yeah, so I'm sure if we were to zoom in on some of these rocks, we would see smaller forms of life. And in the sediment, a type of life called meiofauna. So these sediments aren't barren. They'll have microbes in them as well as potentially meiofauna that sift through the sediment and eke out whatever little organics are in there, which isn't much, and make a living.
Okay, pilots, we're going to go 325.
325, Roger.
Still a little bit of motion in Arctis.
Roger. Yeah, again, we are definitely hearing you loud and clear that the audio and visual is troubled at the moment. Thank you for sending all those informations in. We have passed those along, and it's a little bit out of our hands, but we hear you and are so sorry. In the meantime, if you would like to learn a little bit more about some of the resources available to learn about the monument and some of our work, Kalamehu has some great resources pulled up that he can kind of describe, and this might be a great time to catch up on some of the reasons and the background on the work and the context for it here in the monument.
Yeah, so just so mahalo first to NOAA, to the Cultural Working Group, and all of the other affiliated parties at the monument. I'm over here actually looking at the papahanamokuakea.gov website, and then under there you're going to see a tab that has to do with programming. You can look at the permitting process. You can look at some of the other like specific science and research reports that have been conducted within the monument. There's histories on both culture and heritage and maritime management. There's a lot of information out here for us to intake, and the best part about it is it's in various forms too. So we have a lot of pictures, we have a lot of educational posters for home use and for educational purposes. Specifically, we have like journey books that go through the monument, exploration of reefs, different coloring sheets. There are also a lot of videos that will give you like some cultural introductory cultural context to the place they're in. So we would encourage you to just take the time maybe to like shift the intent and focus, and then to look at these resources just to like give yourself a little more context in that way. Like like I said, a lot of ways to engage. And in the meantime, our OET team is working very hard to work on these issues. So we'd like to thank you guys for your input. I'd also like to thank the OET team for putting in that work 24 hours a day to ensure that we are staying connected in various ways.
For sure, there's a large team all around the world that really helps make OET happen.
One other thing I have to push out right now, if you need to open up a different website, is OET has a patch contest for children 18 and under. Every year we select a patch that represents our expedition, our entire cruise season. So please take a look at that, take a look at the templates available, and we encourage you to get creative and submit your templates or your ideas and designs for next year's expedition patch. Rennie, I'm curious, how do we actually know where Hercules and Argus are at any time in relation to the ship?
Sure, that's a great question and a good time for it. So many people listening may be familiar with GPS that we use on this land surface, and we can use that on the ship. So we know where the ship is via satellite positioning and time of flight. But GPS doesn't work underwater. So what we use for the vehicles is acoustic positioning using sound, the speed of sound through water. Specifically, you might hear us talk about USBL. That's a ultra short baseline acoustic positioning system. So basically, we have a transceiver array on the hull, mounted on the hull of the ship. This is basically a listening array of transducers, and we have beacons on the vehicles. And so because we have ROVs that are tethered to the ship, we're able to send an electrical trigger down to those beacons. If we had, say, submarines or autonomous underwater vehicles, we could send an acoustic pulse that would ask for a response. But in this case, we send an electric pulse, and then those beacons respond with an acoustic pulse, just a sound at a certain frequency. And then we have our listener up on the hull of the ship that hears that and can triangulate its position. So it's bearing to where the ship is and its distance. So we can take that information and basically figure out where they are in relation to the ship. And then from there...
The hermit crab,
Sorry, what was that, hermit crab? And then pair that with a GPS, so we know the actual latitude and longitude of the vehicles. There's a more localized, higher resolution acoustic positioning that we use once we're at the bottom, called DVL, Doppler Velocity Log, and it's a downward-facing acoustic Doppler current profiler. So again, using acoustics, they mounted it. You mount it facing downward on an ROV, and you can use Doppler shift, so change in frequency to assess velocity.
So we could zoom in on this feature here.
On the brittle star?
Yeah, I can't really tell from where I am on the screen.
Sure.
Looks like it might be a coral and a brittle star.
Oh, yeah, you're right.
Thanks for sharing that, Rainey.
Yeah,
Yep.
thank you.
Sure.
It sounds like essentially the ship and the ROVs are playing Marco Polo.
Yes, that's exactly what's happening.
All right, Jeff, I'll hold it here.
Right. Not an enormous coral, but it is a coral.
First we've seen really, I think.
Yeah,
Let's watch.
sorry.
Any ID?
Oh yeah, this is one of the more common ones we've been seeing. Yeah, this is from Muligorgia. It's a Chrysogorgia coral with at least 2 different ophiuroid associates.
Yeah.
Yeah, we did sample this earlier yesterday.
Okay. All right. It was in a slurp.
Yeah, it's
Canister.
the one that was slurped.
Yeah, great. Thanks, Jess.
Sure. Oh, wait, please. It's nice because we're starting to feel the swing in Argus, so hopefully we'll see some more pictures.
Yep. Maybe there's a sponge there in the background.
Oh, yeah. Little sponge.
What is that right there, dead ahead where the lasers are? Is
A
that?
droopy looking guy.
Yeah.
Look.
What do you see? I can't see it on the screen.
It's just to the right of the lasers, right on the lasers, right there.
Oh, yeah, there it is. Looks like maybe an anemone or a sponge, I can't tell.
Or is it
All right, Jeff, go ahead and push on in there.
cucumber? No, it's a
It's like that anemone.
oh, it's another one of those anemones. Yeah. Oh. That's neat. I thought it was our friend Steve, that single polyp. Yeah, no, yeah, Anthomas in any new species is the tentative idea on that. It has bigger polyps. Yeah, and it occurs a little bit deeper, right around 3,000 meters. But this particular anemone was
Right, full wide there, Jeff. Okay,
was sampled earlier in expedition, but we found it a lot deeper, closer to 3,000 meters. So it has it's good to know it has a pretty wide depth range.
nice eye.
All right, we're on the move again.
Yep, headed up slope.
Yeah, I couldn't really see from that angle, but I remember from the last one, Steve, the funnel or the opening was kind of unique.
Yeah, it's puzzling. I sent the image to an anemone specialist, and they are very excited to examine it when it gets to its repository. So I'm hoping we'll follow up with them after that happens.
So in the Argus view, it looks like if Herc stays on this trajectory, there's some kind of white ball thing up in the we're heading towards. See if that's just a shadow or something real.
I guess a sponge.
Yeah, I'm guessing a sponge.
So we have a viewer wondering if a nasty wave or unusual rolls of the ship could result in a collision of the ROV with the seamount and spoil our day. Jake, do you want to take that one? I feel like you're protecting us from that scenario.
Yes, well, I try to stay far enough off the seafloor so that it's a very low possibility, but I mean, a big wave can always come. But right now, I'm about 25 meters off the seafloor, so.
And when you say you are?
Oh, Argus. My position. I'm controlling the wind right now, so I'm about 25 meters off the seafloor. And measure that using this bottom profiler on Argus.
Yeah, so what that's doing, that's pretty cool, is actually decoupling all that wave action from Hercules. So you see Argus kind of going up and down a little bit.
Sponge.
But Hercules being able to stay very steady.
I think we saw the same species earlier on the dive. As a non-sponge expert.
It was actually the first thing we saw.
There, please, Jeff.
This is a, I think it's a hyalinimmatid sponge. Family hyalinimmatid. They have these kind of tulip-like shapes. This one is particularly large, but as we move around, it'd be neat to get an image of the base of it as well. So where that attaches to the rock, if it's possible.
Sure. You want to come a little wide there, please? See if we can find that. Probably from this angle.
I brought my heading around a little bit, faces a little
Roger.
more.
And Steve, this isn't a target
Thanks,
that
Jake.
we need to try to collect.
Chris saw one earlier.
Okay.
So I'm guessing if he wanted one, he would have collected
Yeah.
it. Then.
Okay.
I don't know if we can get a good shot of the base for this guy.
Yeah. You're gonna have to get a little
We get a scoop out for that.
microscope on your manipulator.
Go ahead and push on in there again, and then we'll get cooking, boogie.
Ships come to the top. Arcus has a little bit of swing. Presuming we just want to keep going on this trajectory, unless you have any other suggestions, Beth.
Yeah, we'll keep going. Maybe at the next
All right.
time that the ship makes its final move, we'll sit down and take a rock or something.
Okay. Sounds good.
Oh, right there, Jeff.
Bridge nav. More step 50 meters, bearing 325. Thank you.
Steve, some of the other species of sponges that we've come across have these, there's another anemone, have little crustaceans living inside them. Is that species of sponge known to have that association?
I don't know if sponges have the same kind of associate relationships as corals and associates do.
Mhm.
I don't know if anyone's really looked at that.
Okay.
Well, there are some cases, like some of the euplectellid sponge, like euplectella, have associate relationships with shrimp that live inside the colony. Let's see, what else?
Jess, can you pivot right? It looks like there's a sponge or coral off to our right.
Sure thing.
Yeah, right there. We can have a look at that if we have enough.
Oh, yeah, let's take a look.
But yeah, I don't know if anyone's really looked in great detail outside of those types of shrimp sponge relationships.
Mhm.
You know, some things like some of the squat lobsters we've seen seem to be opportunistically
Okay.
inhabiting sponges.
Is that a coral base there?
That might be another sponge, either a dead sponge base or could be live, sure.
Kind of blends in with the sediment.
All right, Jeff, go ahead.
Looks like a feather plume live sponge, yeah. I'm wondering if this is also the Poliopogon group that we looked at yesterday.
Okay.
We did sample a bit of this, if it is the same.
That little critter there? Oh, yeah. Little skeleton shrimp or something. Nope.
Oh, there's
Nope.
yeah, it's a little single-celled, or not single-celled, solitary hydroid. Yeah.
Wow.
Cool.
Yeah, I think this sponge looks exactly like the one we just sampled yesterday.
Yeah, it does look like what we had in the lab earlier.
Yep.
Okay, thanks, Jess.
Yeah, sure thing.
So is this the base? I maybe you mentioned it, but is the base of that typically not on rock? It looks like that was. Let me see. I have to go back and check which family it was in. wondering if it was a racellid or something else.
Yeah.
Yeah, the way that some of
there's
these glass
many,
sponges attach
I think.
is constrained. It's determined a little bit by their taxonomic organization. Like some of the, I'm not gonna get this right now. Some like the hyaluronamatids have these fibers that extend into the sediment. Often, and the, let's see if I got this right. The plectellids also use fiber, but they attach to the hard substrate, usually.
So they get down deep enough.
Yeah, but they form kind of like a nest on the hard rock, whereas the racelids attach directly using spicules or spicules to the rock. There's none of those like strand, like fibery bundles.
Yeah. Interesting. Saw some cool critters on the way. Was that a brachyngid?
Here's something over here. Can we have a look at that real quick?
Sure. There's an octocoral behind it too.
Yeah, there's also an octocoral. Have a look at them and see what they might be.
Sure. Seems like that octocoral's moving a lot.
Yeah. Seems like maybe this whole time we were just underneath this kind of layer where the started picking up.
Yeah.
May have been, yeah.
They don't like 2,400 meters, but a
Oh,
little.
actually, this is neat. To the left, not this one. No, that's boring. No, that's the Remula gorge. We've seen that before, but the sea-pen here is actually really neat.
Okay.
This is something we haven't seen on this expedition at all.
The one on the left?
Yeah, right down to the lasers.
And I think this might be Cophobelemnon. And there's actually another sea-pen too, right next to it.
Yeah, it looks like there's two of them.
Beluga.
Oh. So this the one that's center screen is the one you're talking about here, Steve?
Yeah, that's the one I want to check out first.
If you want a better close up, we can park it really quickly.
And then there's a black coral to the right of it. So there's quite a
Yes,
bit of
please,
diversity.
Jess, park.
All right, go ahead and go full wide for a sec there.
I don't think
We've got space here.
I don't think there's any sampling intention with these, but it would be neat to just image them.
Yeah, especially it's the first time we're seeing it. Get a record of it.
Yeah.
All right, sit ourselves over here. There's something else right in the center, like really low down. You see that? Is that a stock crinoid or?
Yeah, I see what you're talking about. I can't tell from this angle.
That's an Beluga.
Right,
That's the Beluga.
Oh, that okay.
The sea pen right here is the one that's sediment, and then to the right of it is a different black coral that actually looks like alternative apathes. So, but it's very, very small.
Good.
So that would just be an approximation at this point.
Wow.
Yeah.
Very cool.
I don't think
Neat
I've
little
seen that one.
sea pen.
Very cool.
All right, we might have to
I think
pick up
this
in a
is in the
moment.
genus Cophobelemnon.
Do we need to get any closer, Steve? Is that good?
No, this is wonderful.
Okay. Thanks,
Actually-
Jess, for making that stop.
For sure.
We need to get a move on.
Full wide, please. Thanks, guys.
That's the first Beluga I saw on this expedition, unless other watches they've been seeing them.
Yeah, other watches I've seen a couple.
It definitely seems to be getting steeper. Some really beautiful basalts up ahead.
I'm just gonna push up in front, and then we'll have plenty of time for some more
Yeah, you can
close-ups.
fly up there.
Yeah, if you're just joining us and noticing that the broadcast video is having some issues. We do apologize. We are aware of it and pass that along. Probably, yeah, unfortunately, our telepresence is not without vulnerabilities sometimes.
Yep.
With that said, I think we made this announcement before, maybe as another activity, we'd encourage you guys to hit up either the OET websites or one of the two main websites that can provide some cultural context to, multidisciplinary context into Papahānaumokuākea. So for instance, on what I'm looking at right now is papahānaumokuākea.gov in the About tab, and it just gives us an explanation of the name. And so there's an opportunity there to, with a soundbite that assists with the proper pronunciation of the monument, as well as to an audio description of the name. And so maybe in the meantime, there's also a map. There's also a map over there of the monument itself. And so we would encourage you guys to, in the meantime, pass the time, maybe to familiarize yourselves with the monument, and then, like we said, we like to acknowledge our team over here at OET, specifically on Nautilus as well, that it's working very hard to ensure that these issues are addressed. So thank you for your comments, and yeah.
And thank you for watching.
Yeah, we still have almost 50 unique logins from around the world. That's great. Thanks for sticking with us despite the low quality visuals.
So that ship move is complete. We have approximately 20 meters of swing on Argus. Did I hear you say you wanted to pause at that point and kind of look
Yeah,
on?
yeah. We're getting to an area where we can sit down and not be pulled away. Maybe do a little rock hunting.
Ooh, very nice. Yeah. Might be a nice feature over there, right?
Yeah.
Yeah, the one on the left was a bit like where we were seeing stuff at the base of it. It looks steeper, and then once Argus ascended, it kind of flattened right out. So
Just can we have a look at this feature over here?
Oh yeah, sure.
I guess maybe just a sea star.
Yeah. Good. Put it in there, Jeff.
They deserve some quality close-ups as well.
Oh,
yeah.
Sinjid. Sorry. Sorry. Change this.
Got its arms up in the water. Wait for a snack. All right. Thanks, Jess.
Thank you. Sorry about that one. Not
No,
very good.
it's good.
Pull away, please.
No. What is this?
All
Shrimp.
shrimp. All shrimp.
Oh, okay.
Steve, do you know if there are any deep sea organisms that use a web hunting strategy similar to a spider?
One away, isn't that kind of what spiders are?
Yeah, that's one thing.
Or like salps?
What about larvae?
Yeah. That so tunicates, I guess, could be construed as they have a mesh basket within their anatomy that allows them to filter water and trap particulates. I suppose you can make an argument that fly trap anemones might do something similar too. But in terms of like structures that are produced that like are persistent, and the animal comes back and checks them, is that, yeah.
Steve, what about those snot houses, the
Think about
larvaceous snot houses?
Yeah, yeah, like the appendicularians. Yeah, those are midwater organisms. That would be a pretty good example.
Oh, there's a shrimp in Argus's
5
camera
seconds.
every now and then. There it is.
There it is.
Larvaceans or appendicularians are a pretty good example of animals that secrete a mucus, not so much a net, but a mucus that traps marine snow, small plankton, things like that. And then it will consume the material it's trapped over the day, discover their house, and create a new one.
Oh, there's a little thing right there. If we have a look at that.
Yeah, sure thing.
Very hard in this.
Oh, there's a
hard
fish
to
too.
see small things.
Oh, yeah. Which would you guys prefer, the squirrel or the fish? Or both?
Yeah, maybe we can do both if we have the time.
Yeah, we have.
Yeah, I guess it's just about settled where,
Okay.
where it will be.
Great. Yeah, let's have a look. Oh, and there's also something right there.
Oh, yeah.
All kinds of things to look at. Yeah, we can start with this one.
All right, go ahead, Jeff.
Is that a black coral?
It is.
Do one more bump up here.
Yep. This looks like stauropathes. It's a little irregular shaped. Might have damaged at one point, perhaps. Yeah, stauropathes. One we've seen a little bit on this dive, and in previous days.
All right.
Pretty common.
All right, pull away there, please.
See if our fish friend is still around.
Oh, yeah, still staring at us. Would it be possible to get more of a side view of this? Sure.
All right. Who remembers this fish?
It is a
yesterday.
Trompopsis.
Tron.
Strontops.
That's it.
There you go.
Rennie beat me.
And you were saying yesterday that this is a type of angler?
Angler fish.
Yeah, so I'm wondering if we can see this little lure. All right, Jeff, you want to put on there, please? Surprisingly, doesn't seem very perturbed by this vehicle.
Doesn't look like much bother this guy.
The name like Troniclops, I mean,
Tronoclops. You don't have a lot of natural predators with a name like Tronoclops.
definitely not.
It's like, look at it. It's like holding on to the rock.
Yeah, it does look like it.
Yeah, like
Wow.
What an interesting texture to this
Yeah.
skin.
Yeah. I don't really see the lure.
I don't either.
It's right between the eyes. There's a small tan colored
Sorry, checking a
semicircle.
little bit of
So it's just the lure. It's not actually protruding from the head.
It's not protruding. It's
Yeah.
tucked into a little crevice
Yeah.
between its eyes. Yeah.
And probably if the lights were off, you would see some glowing or something like that.
Oh,
We've tried, and it never shows up well on
Yeah.
HD.
Need some low light cameras for that.
oh.
All right.
Alrighty.
Thank you.
Yeah, for sure. Full wide. I didn't swim away.
So lots of rocks here. We were just discussing that about 70 meters to the north.
Mm-hmm.
You can see in Argus there's possibly some larger. Here, I'll put my cursor on it this bit here.
Yeah.
How far away did you say that was?
Looks 70 meters, and Argus is 315, so that looks like north. Due north 70 meters is that, but I don't know its prominence. We just know that Argus is picking it up at least.
Yeah. We would need to put in a ship's move to get up there, correct?
Yeah.
Okay.
Oh, it's swimming.
Oh yeah. Bye, fishy.
There's
Oh,
yeah,
I see.
winch.
Sorry.
Another, sorry, there's a shark and winch cam. Wire
Oh
cam.
yeah, shark and winch cam.
Oh,
Super
yeah.
cool. Two of them.
Yeah.
Where? Oh,
No wire
good, I
cam.
see it.
Yeah. I'm guessing they're sharks.
Let's see. I was actually just thinking when we're looking at that fish, like it's almost like the fish is telling me to pick up that rock, hold onto it.
The guardian.
Yeah, so this. I'm trying not to be too picky about the rocks so that I get kind of a random sampling. Actually, if we could just sit here for a moment and
sure.
look at the rocks and see how altered they are.
Sure thing. Just anywhere around here?
Yeah, maybe let's look at these.
Roger.
That's a pretty interesting observation, Beth. Yeah, just from the cultural perspective, is like, the fish is there. I don't know. I wouldn't necessarily know if they chose that for a reason, but why not follow our friends?
Yeah.
Little coral there.
See that 5 function is
Yeah, a little bit of
angly.
rock.
Yeah, so there's a coral there on the right side. Okay, let's see the
pretty
lasers
low.
here.
Jake, you want to move Mongo up a little bit? Yeah. Get him on the dirt. I think you might just need to rest up or... Yeah.
Really just bouncing right there.
I can back off so you can get some better range.
Good view of one of the knives we have on the vehicle, just in case we encounter Chronoclops again.
Thank you.
There was some knife action going on the last dive, wasn't there? I remember coming into the video and seeing a knife being moved around.
I think that was for training, yeah.
We have those for safety in case we encounter any fishing lines or
Yeah,
Yeah, maybe
other
elbow
ones.
up a little bit.
it's always a
and
good
then
idea to have.
wrist over a little bit
All right.
to have dirt.
Spicy.
Very spicy. You're just resting, right? Nice. Nice. Maybe just double down. It's on the bumper.
Jeez.
It's really spicy. We should probably toggle those.
sound is clicking. It's not even a second.
Maybe elbow up a little bit more there.
Right.
Just play with it like this for a while.
Wrist down. And the wrist is slow movement.
What kind of coral is that, Steve?
This is another of the Remulogorgia corals. The common Chrysogorgia golden coral. It's by far one of the most common corals we've seen the whole cruise.
Mm-hmm.
Yeah, they this particular genus, during the capstone expeditions from 2015 to 2017, it was one of the most commonly observed corals in this part of North Pacific, so the Monument, Hawaiian Islands, even further north towards the Musician Seamounts. However, when you move further south, it tends not to be as prominent, and you typically find members of this genus deeper and in higher densities, but only on, or not only, but primarily on vertical surfaces. They like to form on walls.
Did you see that little jumper?
Yeah. What was that?
Was that a tiny squat lobster of some kind?
I don't know. Yeah. Oops, sorry guys. It moved,
Yeah,
like a little
flop down.
cephalopod.
Any more?
Yeah, you can. Yeah, any more zoom on that there, Jeff?
So small.
Oh no.
Yeah, what is that?
If we got any closer, maybe we'll find out what it is. My eyes looking from the back row to the front screen, and
Yeah,
I can't make it out.
Yeah.
How are these
Could be a
rocks
small
looking for you
crustacean.
guys?
Yeah, can you pan the camera?
there it
Oh, there it goes.
Oh,
Yeah, it looks like a squat
squat
lobster.
lobster.
Yeah.
Well, lobster.
Totally a squat lobster move.
All right.
Dive bombing.
2 animals. That was me. This is the rock I should pick up. Yeah, so just that rock that you were originally looking at. This one right here.
Oh,
Is that too big?
that's pretty big.
Yeah. Okay, I can't see the lasers. Oh, yeah, there they are. Okay, yeah, that's probably like 30 centimeters. We don't need that big. Can you zoom in just a little bit, Jeff, please?
There's one to the lower, to the kind of mid left there, or any of those. Those are more manageable, or the ones kind of center screen behind the coral.
Yeah, like one of these.
Yeah.
Is that what you're talking about?
Yeah, or the one center screen behind the coral too. Those might be better bite size for us.
Yeah, okay. Let's try this one and see what it's like.
Okay, yep. See how that one goes. This one might be a little bit bigger sample. Is all right.
As long as it fits in the forward box, right?
Yeah, it
Forward
would
box.
go in the forward box.
Roger, you want to come full wide there, please, Jeff, for a moment?
I don't think I've ever seen a squat lobster move. That was pretty cool.
I don't think I have either. Not quite like that.
Yeah, they'll shoot backwards
Okay.
when, especially if you go up with a slurp gun and you try to suction them, they'll usually escape that way.
Very
So
fast.
you kind of have to be smarter than them by
Going behind them.
going behind them.
Yeah.
Doesn't always
The
work
first
out.
time I saw coming from Maine, lobsters are super popular. The first time I ever saw one move in the water, like getting away from
Good,
something,
there,
was
please,
mind blowing. Yeah, same thing, right? And they're like just jet backwards.
That's great.
It's awesome.
Just to confirm, it's going to be this guy beneath
Yeah,
the
it
jaws there.
can sometimes be terrifying
Yeah, that one right there.
when you encounter a lobster on a scuba dive or something.
Yeah, of course.
They can get quite large. Usually they're in holes and stuff like that. It's probably the safest place for them to be.
More in there than I expected.
Oh, was it attached?
No, we'll get it.
Oh, yeah,
Over.
you got it.
Let me get a better grip on this guy.
Nice.
All right. Can we get our little glamour shots, please?
Yeah. Go ahead, Jeff.
All right. Yeah, this looks like it's going to be somewhat similar to what we've been getting. A little manganese rind, but it's probably not going to be very thick. Surprisingly.
It looks exactly like rock this morning. It's very hard to tell them apart. They all have this very smooth around the edges where the edges of the sediment are is very smooth, but underneath it's more granular.
Mm-hmm.
The top has sediment on it.
All right, so in the toolbox, is there any floaty bits that we need to know of?
There's nothing in there right now, so you can make your choice.
Cool beans. All right, flow wide, please. Jake, you want to go ahead and pop it up, please?
All right. My squat Chanaclops rock.
Yeah, and if you don't mind following me in a bubble there, please. Yeah, you on it. I'm telling things already know.
What's the sample number?
Sample 47.
047.
It's nice to have the old controller back.
All right, great. Thank you, Jess.
Yeah, no worries.
Water.
Yes, I'll need to take a Niskin.
Oh, yeah, let's do it. Sitting okay, or?
Yeah, sitting is fine.
Roger. Niskin. Is there any way to view the Niskin top?
Yes.
Top nose? Right.
Have we heard anything from our viewers about whether the satellite feed is improving?
Not in the last 6 minutes.
Okay.
I just turned it off. There we go.
So for our viewers at home who maybe haven't been watching other dives of this expedition, we have multiple goals for the scientific objectives of exploring these seamounts in the Papahanaumokuakea Marine National Monument. One of them is to understand how the rocks that are exposed on the seamount get altered and are sites for minerals to precipitate, as well as microbial life that is
Number 6.
number 6. Great.
Come on.
Microbial life, microscopic life
Here's
that
that
is in
sample.
these rocks.
There it is.
There it is. Yep.
We're collecting the rock samples. We're also collecting a corresponding water sample from where those rocks are taken so that we can understand what the microbial community is like in the water that these rocks are bathed in. So that was all of the sample we just took there. Paired samples, I guess.
As you can see, the 5 tops that are still open, they're pulled open and then held by a spring-loaded, or I guess the spring is pulled back.
Yeah, right. So there's a band connecting that top to the bottom, and then there's little ropes on the back that are holding it open. And then when we pull that lanyard, it releases a pin and lets that rubber band pull the tops closed.
Is there anything else you want from here, or should we?
No, I think we're good. If we can put in that ship's move that Rennie was discussing, I think now is a good time to do that.
Roger.
Sure. Let's go north and see. It's interesting.
Roger. And then we'll have you stow Mongo
Yeah.
to the side.
Other times on the dives we're doing on these seamounts, when we do find areas of high coral density or animal density, we also collect water samples, and that is for a parallel study of how animals shed cellular material into water that includes some of their DNA. And so we can use those water samples to identify the animals without having to necessarily collect the animals. So we might collect some of those on this dive as well, if we find some good animals. Well, all animals are good, but a high density of those animals.
All right. The satellite, or sorry, the stream is still pretty choppy visually, but for the most part, it sounds like folks are able to hear us or make out the audio, even though it is still cutting in and out a little bit.
We've got a shrimp in the screen.
Is that a big shrimp?
That's a really big shrimp.
It's a really big shrimp.
Massive shrimp.
Can we have a look at that?
Yeah,
That's
jumbo
like 20
shrimp?
centimeters long.
Like, yeah.
So for the viewers at home with pixelated video, we're passing over kind of a rolly part of the seamount, and there's a at least 20 centimeter long crustacean swimming around very beautifully.
Jumbo shrimp.
Seems like it's on a mission to get somewhere.
Oh. Little swimmerets or pleopods. So graceful.
It's a cool swimming motion too.
Yeah.
See, there's another small
yeah.
shrimp.
Oh, just for scale, yeah. There you go.
That's a great shot. Thanks, Jess. Yeah, sure thing. And Jeff.
Two species of shrimp, nomadic arsinid in the right-hand side with the long legs, and then I think this one's called Aristiopenaeus. It's a swimming shrimp.
Very pretty. It definitely is.
All right, full wide there, please. Thing in the upper left corner there for a second.
There's a sponge up there, yeah?
There's a sponge.
Again, for our viewers at home, so we just put in a ship's move so that we can continue on our transect. So the ship is slowly moving and bringing Argus along with, and Hercules swims along underneath. And we're headed towards a feature a little bit to our north that is showing up on the sonar as maybe being a taller feature. And we're coming into view of a sponge. Doesn't seem to be attached directly to a rock, maybe. Kind of in a cemented little pool. Lots of little rocks in this area, but more sediment than we have been seeing.
And just as a reminder for some more viewers that might be experiencing some technical difficulties, we've said before, right, there are a couple of websites that give us a little more context into Papahanaumokuakea. So a site that I'm looking at on my screen right now is one that's done by the Office of Foreign Affairs, one of the co-managing parties of the monument itself. One of the things that we can look at, if you want to look into some of the context of the management strategies that are incorporate multidisciplinary things from scientific best practices to also cultural traditions and thoughts. A report is called Maikopome, and the intent of this document is to establish collaborative management framework that guides the cultural agencies towards integrating traditional Hawaiian knowledge systems, values, and practices into management. So we would invite you guys to just check out the various resources that are available. There are a series of multimedia resources as well. So we have cultural briefing videos, some sound bites of some of the protocols that are taught for specific purposes for these expeditions, as well as a lot of fun, interactive, educational resources that you might use if you're an educator in your community and you'd like to find more ways to access information about the monument to pass on to your classrooms, or just do it in the downtime while we wait. So, and then once again, go check it out. Mahalo again to our team behind the scenes, working very hard, especially to get the feed going back up.
Thank you for that, Kalamayu. And we're continuing our kind of northwest, northish transect. Looks like we're coming out of an area with some nodules into an area that doesn't have as much noticeable extrusive basalt. It's much flatter here.
It's a really distinct line there.
Really
The
distinct
Argus
line.
camera.
Yeah, so almost like a slide event. Lots of little nodules and a sponge coming into view.
So extrusive makes those boulders. What would be an example of intrusive?
So some of the ones we've seen on other dives where we come up and there's like big blocky features, those are typically more of an indicator of an intrusive injection of magma.
It's like a whole field of these sponges, but
yeah, so it seems that their sponges seem to like these nodule fields. There's a lot of them in the, relatively a lot in the view from Argus. Jeff, if we could get a zoom in on this one coming into the center of the screen, just to see if it's the same thing we were just looking at.
Yeah, I think it was.
sure thing.
And I kind of tempted to scoop up some nodules and put them in the box.
Yeah, you are. Do it.
A push core.
Yeah, we can try a push core. Let's try a push core.
I think I'm more tempted for this soup.
Yeah, I know.
All right, go ahead and push on in here, please.
That looks pretty similar.
Yeah,
Looks like there's a tiny little star attached to the bottom.
in our view, it looks like there's a number of these
Yeah,
scattered.
quite a few. Yeah.
I'm not sure about my identification strike now. I'm not sure if it was a ferronematidae or something else. To go back in my notes. But yeah, this is the same, exactly the same texture we collected yesterday.
Yeah. All right, Jess, dealer's choice if you want to try the scoop or the push core first.
Rather full wide, please. Let's try the scoop. We'll have more success with that.
Okay.
Let's see if we can get a little ahead,
Yeah.
just in case. We are moving towards that. So it's now 60 meters out.
Oh, looks like we got plenty of.
There's any to the right
Yeah.
of this manganese stuff.
Yeah, it looks like there's pretty extensive fields. We'll
Yeah.
just go to the end of our tether as long as we're on this field.
Yeah.
Yeah.
See some of these other sponges while we're at it.
Oh. Follow the sponge brick road, as they say.
So for anybody at home who's
That's okay.
seeing just pixelated views of this, these nodules are probably pretty small, on the order of 1 to 2 centimeters or half an inch to an inch in diameter.
Give me a little bit
Little
of
black pebbles in
leash there.
very close proximity to one
Yeah.
another, with some sediment underlying it.
Is it a little grab?
Yeah, so we're settling down in an area. There's a sponge off all around us. Looks like we've gone to the northernmost edge of this little slide feature, but we're still in these nodules. Settling down to see if we can take a sample.
I hadn't really sampled or held nodules until just a couple of days ago, but they get a really different weight from the other basalt rocks because they only have a small amount of the, maybe sand or basalt, but yeah, they're very lightweight.
Yeah, they definitely have a different density than
Jeff, you want to
basalts.
go ahead and push on in those nodules real quick? Nice. Jake, would you mind showing me that arm? Thank you.
Yeah, and now that we're on this edge, it seems like there's many sponges maybe off to our right, but I don't think we're going to get any luck with push core, so
Full
Let's
wide, please.
just try to scoop.
Yeah, so again, Where is the scoop? partial conception here about the connection with the lava activity. These were not formed in the original, in any
No.
original eruptions.
I don't see it. Is it in a box?
I don't see the scoop.
Oh, do we,
Oh,
are we scoop this?
Did they put it in the starboard box?
Oh,
I don't have any notes about containers in the box.
No, we didn't put it in the starboard. Let's see here. Jake, you want to pop out the toolbox?
Yep.